So I'm Jessica Damoiseau, and it's my great pleasure to introduce the last keynote speaker of this meeting. Some of you may wonder how to pronounce his last name, or actually his full name. So let me help you with the correct pronunciation. So it's Martijn van den Heuvel. So listen carefully, I'll repeat it for you. Martijn van den Heuvel. Easy, right? So I first got to know Martijn when we were both undergraduates working in the same lab in Utrecht University. We had a good group of very enthusiastic undergraduates, but one person clearly stood out. Although we all thought we were a pretty smart bunch, we quickly agreed that Martijn was really smart. But it was not his intellect that impressed us the most. It was the quality and quantity of the questions he asked. Even to Carl Friston during our first scientific meeting out of the lab, while the rest of us were too starstruck to speak. We were deeply impressed. Martijn has now risen to stardom himself. His extensive work on brain connectivity using graph theoretical approach greatly impacted the field, as evidenced by the more than 11,000 citations over just the past 5 years. But students, don't feel starstruck or hesitate to approach him for questions, as Martijn is one of the most humble and down-to-earth persons I know. I know a lot of us look forward to hear us speak, so without any further ado, help me introduce Martijn van den Heuvel.
Good morning, everyone. It's great to see you guys here on the last day of, I think, a fantastic OHBM here this year. It is a great pleasure and a great honor to speak to you guys here. And special thanks, of course, to Jeske and the other members of the program committee for being this brave to invite people like me on the stage. So this morning, I would like to tell you something about that I like to call the principles of wiring of the human connectome. What are the underlying fundamental principles that govern the complex organization of the connectivity of the human brain? And with that, how might they actually play a role in shaping behavior of the human? And perhaps, as I hope to argue in my talk, how might it actually also shape a landscape of abnormalities that may occur and perhaps may not occur in the human brain? But before I begin, I would like to tell you a little bit of a story. So let's assume for 1 minute that you're not in the field of neuroscience. You're not an MRI expert or a neurosurgeon or a neuropsychologist or a neuro whatever. You are a clockmaker. And one day this guy walks into your office with this old pocket watch, right? And he tells you that it's of his dad who passed away. So it's very dear to him, and to honor his dad, he wants to start using it in daily practice. However, the clock is broken. Well, you are the best clockmaker in town and a little bit of a sentimental person, so you take on the job to fix the clock for this man. What is it that you're going to do? Feel free to shout at me. I'm Dutch, so I'm used to it. Take it apart. Take it apart, exactly right. That's normally what we would do, right? We just first observe, and then we're going to take the clock apart and take out all of its elements. I already prepared the slide because, from my experience, neuroscientific people are very predictable. So the aim here is, right, we have a complex problem and then we reduce it into smaller problems, sub-problems. And then the idea is that if we understand every one of the individual elements, that we then have a better understanding of the total system. This is what we call the field of reductionism. It's a very fruitful approach in physics and math and also in neuroscience, right? We're learning a lot about the individual parts of the human brain. But clearly we're also missing out on something, because this pylon, radius, and springs is not automatically a clock. What is it actually that we're missing? Relationships, right? Exactly, right? We miss out on how every one of the individual parts work together, right? This is the obvious one, right? This is, you've all could have said this, right? So, shall we give it a try? As a starter up? Say it all together? Humor me a little bit, right? Shall we do it? I do 3, 2, 1, and then we all say connectivity. Right? Shall we give it a try? Give me a hand here, right? Okay, let's go. 3, 2, 1. Awesome, right? That felt good, right? Let's do one more. And now we do network, right? Okay, just go one and then we really go out, right? All people are gone, right? So there are no supervisors here, so we can give it a try, right? It's 3, 2, 1. Awesome. My job is done. I can go home and tell my daughter that everyone at Human Brain Mapping talked about connectivity and networks. So the anatomical infrastructure for all of the communication and integration and communication and collaboration and relationships between the different elements of the human brain is of course what we would call the connectome. Like your genome is a full description of your genetic information, the connectome is a description of all of the connections that build together the nervous system. Well, the goal of the field of human connectomics, and with that the goal of my lab and with many friends in the world, is to build as good as we can a map of the human connectome, like the roadmap of connections, and then study this map through means of network science, and study this map to get a better understanding of the underlying fundamental principles of how the wires in the network are placed. And in particular, we're interested in getting an understanding of how this layout of the wires actually plays a crucial role in shaping brain function. The underlying principles of wiring. So one way to do that, one way to measure the human connectome is of course by means of MRI. And we're actually very lucky to be able to be working with this technique, because it's one of the techniques that actually very uniquely gives us information about both the structural connections and the functional connections of the human brain. Think of that, right? There are not many other methods that can do both. Now, there are, of course, however, quite a bit things to say about using MRI to measure the connectome. For one, we're not measuring connectivity on a neuronal scale, right? Not at all, right? We're measuring and reconstructing pathways and functional interactions between large-scale brain regions. And for that matter, we're actually mostly dependent on derivatives of the signal, right? So in diffusion MRI, we have the diffusion of the water molecules that we're measuring, which we then use to derive or estimate the pathways. And in fMRI analysis, in functional connectivity analysis, we're heavily dependent on statistical analysis. And for that matter, we are among friends now here, so we could say that sometimes MRI is a little bit noisy, right? And this can have an effect on the reconstruction of false positive and false negative connections within the network. And actually, last year, Andrew Zaleski published, I think, fantastic work on showing that these false positive and false negative reconstructions can actually have a quite severe effect on your network topology. And together we published a follow-up paper showing that this can have, in turn, quite severe consequences when you do a case control study. So for example, if you're in the field of disease connectomics or basically studying any type of group and you want to measure or compare your network topology between groups or across age or intelligence or whatever metric you have, just go see these papers, because it can give you a feeling of how to deal with this type of biases in network topology examinations. But on average, I'm a pretty positive person, so I like to see the glass half full here. I always like to compare the maps that we make of the human connectome today with the maps that we had in the world in the 17th and 18th century. At that time, we only have these type of maps, right? They were pretty coarse, with potentially many false positives and false negatives. But at that time, they were extremely useful to do many new great discoveries. And I like to see the connectomes that we have today in a similar fashion, right? Yes, of course, hopefully in years to come, we will have better and better technology and getting higher and higher resolution. But today, there might also be great to do new discoveries. All right, so for the rest of my talk, right, let's put aside these potential problems, but let's assume that we have the perfect technology available to measure the human connectome. Right? What would it look like? Well, probably something like this, right, like a bowl of pale spaghetti. And that's not even a joke. It wasn't. No one was laughing. But if we look at the neuronal scale, it looks like this. And if you zoom in and you use diffusion-weighted MRI, it looks like this, right? So this is pretty much brain spaghetti to me. So the point here is that we need another layer if we want to dig deeper and want to go beyond the study of one individual brain region or one individual tract. And we want to get a better understanding of the topological structure of the network. And one way to do that, of course, right, is graph theory or network science. Well, it doesn't need much introduction, I think, but Basically what we're doing is we're describing the brain regions, could also be neurons, right, as the nodes of the network, and then the relationship between those nodes that we aim to reconstruct by means of synaptical reconstruction or fiber bundle reconstruction or correlations as the edges of the network. And once we've done that, we can actually use the great toolbox of network science to extract all different types of topological features of these networks. We can look for whether some regions might be more centrally embedded in the network. Might look for how the network is capable to facilitate information integration between different areas of the brain. We can look for whether the brain actually might have sub-network formations, modularity as we like to call it. Or even we can then again look at which type of attributes might bring integration and communication between those modules. So now how can we use this toolbox? Now we have both ingredients, right? We have a good map and we have the tools to study it. So let's dig deeper and search for our main goal of the morning, right? Find the principles of wiring. Now one way to do that actually might not just be to only look at the human brain. And there are, of course, I think in the last 5 to 10 years, ample examples of studies that have started to look at the human brain. But perhaps we should not look at the human brain only, but if we really want to look at these underlying core principles, perhaps we should just start looking at a wide range of species. Because the idea here is that if some fundamental principles have survived millions and millions of years of evolution and evolutionary pressure, then these might give us like a good clue to the really fundamental principles. So the field of comparative connectomics therefore describes, or aims to describe, both the commonalities and the differences in the wiring organization across species. And in a recent study, Olaf Spoons and Ed Bromer and I, we started to combine as much information as we could from the last 5 years on every one of those studies that studied these connectomes of every one of these individual species, ranging all the way from the worm, neuron reconstruction connectome, 302 neurons, 6,000 connections, to the reconstruction of the fruit fly brain, to the mesoscale connectome for rodent species, and to the macroscale track tracing of several mammalian species, including cat and several primate species, and of course, in the end, also the human brain. And the first common theme we observed is that all of these networks are relatively sparsely connected. And there's a very simple reason for this, right? Making connections is really expensive. Maintaining it, you have to build it, you have to maintain it. It takes a lot of energy and neural resources. So neural systems want to keep the amount of wiring as low as possible. And second, it also has to fit in a certain limited amount of space, right? So there is a very strong spatial constraint to this. And Daniel yesterday clearly showed this. So there's a strong spatial component to this. And this might have given rise to the formation of and the favoring of particularly short-range connections, and the formation of locally connected modules. Now, the formation of these modules in turn might have given an infrastructure for specialized brain processing. For example, if you look at these modules in the fruit fly brain, you can actually clearly see that they overlap to known functional systems of the fruit fly brain, right? Like motor system control, visual auditory processing, etc. And the same holds for the human brain, right? If we look at I think in the last year, seminal work on finding the different resting state networks of the brain, we can interpret them as these functional modules, and every one of them actually overlaps to known functional domains of the human brain. And even more, they also overlap with known anatomical tracts of the brain, suggesting that there's a real strong anatomical wiring basis for these functional systems to emerge. However, the drive towards parsimonious wiring and the drive to forming strong spatial constraints, and with that perhaps spatial gradients across connectivity, could not explain the entire level of complexity that will be observed. So here too, across all of these species, ranging all the way from the neuronal level connectome of the worm to the hopefully more complex network of the human brain, we also found that these networks tend to have a lot of neural resources invested in features that might bring topological integration. And these include short communication paths, centrally connected hub regions, a centrally connected core, a rich glob in the overall system. And the argumentation for this is that although these features are extremely costly to build, they might have an extreme benefit for global brain function. So these neural systems, right, although again it's very costly and they have to reserve a lot of resources to build it, the brain function, the evolutionary pressure to increasing brain complexity and brain functionality might be a strong driving factor behind this. So for example, in the human brain, structural and functional studies have clearly shown that there are many centrally connected hub regions in the brain. And interestingly, when we closely look at their spatial location, we can actually see that they're almost one-to-one overlapping to known association regions of the brain. Regions that for decades have been suggested to be functionally important in terms of integration of information. And these studies now show that there's like a real hardcore wiring background behind this. Even more, when Olaf and Osporns and I a couple of years ago, we started to play around with the idea, then how, if we look at every one of these hubs, right, we just see them as almost independently, right? We compute the degree or between the centrality or participation coefficient, but that's almost node-based, right? But how are these hubs together or then connected? And what we noticed is that these hubs actually have a disproportional number of connections between them, much more than you would expect if you would just look at the overall connectivity degree sequence of the network. Suggesting that apparently on top of the already expensive hub regions and expensively rich regions, they also start to actually form a club together and actually have ask for even more investment in terms of their interconnectivity. And we found these hubs to be distributed across almost all of the different functional network, not just the default mode network, not just the salience network, but we actually found them across all different functional domains. This has been verified now in the C. ligands worm by Emma Towson, for example, but also in the fruit fly brain, beautiful work here by Shi and colleagues. So this infrastructure, this rich club might potentially really be an anatomical wiring infrastructure for neural integration across the system. Even more, we started to look deeper in this, and we said, well, but this is all mathematical modeling, right? In the end, graph theory is math. So how does this relate to the hardcore biology? And this is ongoing work of one of the postdocs in the lab, Liana Scholtens, who's doing beautiful post-mortem work now, in which we aim to just zoom into those hub regions and look for their neuronal level of organization. And what we observed first in the monkey is that actually the degree distribution across the cortex almost one-to-one overlaps to the neuronal level of complexity, right? Suggesting that these hub regions are not only rich on the macro scale, but also on the micro scale are heavily rich with neurons, with complex dendritic branching, a lot of synaptical density, like really suited in that sense for neural integration. We later verified this in the human brain through means of post-mortem histology again, but also more recently, another student in the lab, Yongbin Wei, looked at the big brain data set and integrated that with ACP connectivity data. And there too, we noticed that the gradients of connectivity actually relate to the gradients of the cortex in terms of their complexity again. Again, suggestive of these hub regions to be highly suited for neural integration. Furthermore, others have also looked at the genetic underpinnings. Beautiful work done here by Fonito and colleagues in Krinen, showing that actually that perhaps genes related to metabolic energy use and neural formation might be potentially expressed and co-expressed in these regions. Now, if it is true, right, that these hubs in the rich club play a crucial role in shaping brain function and integration, we might also argue that damage to this system might have a disproportional effect on brain functionality. And one of the disorders, actually, that has long been hypothesized as a disorder of brain disconnectivity is, of course, schizophrenia. Actually, schizophrenia means the split of the mind, right? Not the Hollywood term of two personalities, but the word actually says this, a split of the mind, like already suggestive of decreased integration between different brain regions. And if we look at modern-day connectome science work, these studies actually too pinpoint in a direction of potentially these hubs to be disproportionately affected. Very nice work here done by Danny Bassett and, of course, the group of Ed Bilmore. And later on, we also give it a try by using diffusion-weighted MRI to pinpoint these hubs. And what we particularly noticed is that the regions that tend to be the most affected in terms of their network properties were also the ones that actually had the highest degree and were potentially the central hub regions of the brain. So we then looked further and said, well, let's look at this central rich club structure in particular. And one thing we can notice there, if you look at these connections that span between the rich club nodes, we tend to call them the rich club edges, that the disease effects there are disproportionately larger than compared to other roads and other connections of the network, like the feeder and the local type of edges. We found this in patients, but also in their siblings, suggesting genetic underpinnings of the effect, as well as more recently in high-risk subjects, suggesting that these effects might potentially already be there before the onset of the disorder and are very likely to be a neurodevelopmental effect. Here too, we looked a little bit deeper and looked at the neuronal scale. And here too, because one of the hallmark features of schizophrenia is a reduced synaptical density, right? So we correlated the disconnectivity profile across the cortex with, on the macro scale with the disconnectivity profile on the neuronal scale, and we found these beautiful correlations. And even more, when we, inspired by the work by Fornito and Krina, when we looked into the gene expression level, we noticed that if you just simply take the schizophrenia genes, right, resulting from a large GWAS study by the PGC, and then you look where these genes become expressed in the cortex, right, using the Allen Human Brain Atlas, these regions that show actually the highest expression were also those regions that showed the highest level of disconnectivity in the disorder. Again, really suggestive of a strong genetic underpinning of these effects. So taken together, so far we have seen that we can look at these commonalities, and there might be two of them, right? At first, a strong drive to minimize wiring, and the other hand, a strong drive to put a lot of effort and energy into forming network attributes for topological integration. Now, it is important to notice that these two features are not independent from each other. Actually, they're almost opposing forces. So if we would put them on a dimensional space, they actually span a complete area or a morphospace of possible network configurations. So on one end of the space, the yellow ones, we would have networks that take a lot of wires and a lot of complexity. So in that sense, they're extremely expensive, perhaps too expensive for the neural brain to handle. So these would be biological implausible networks. On the other end of the spectrum, the green ones, we would have networks with a relatively simple topology, not that much wiring, and perhaps as such, not able to support all of the complex brain functionality. So these two features, right, they're continuously betting for resources, so they can only be mutually joined in terms of a trade-off between them, which is the blue line. And we argue that the human connectome is hovering around this optimal trade-off. Now, as we can use comparative connectomics to start looking at variation in connectome structure, right, we can actually also look at in commonalities, we can also look at how variation in connectome structure between species might perhaps form a foundation for variation in behavior. And I think this is a particularly sexy thing to do, right? And there are not many studies out there, unfortunately, yet. So I urge you to start doing this. But I found a few gems, and I think they're really worth to highlight. Now, our first one is, I think, a fantastic paper by Boombacher and colleagues in Cell a couple of years ago, in which they reconstructed the so-called fairing system of the C. elegans worm. So the fairing system is the eating behavioral system of the worm, and it is a subsystem, and it contains around 25 different neurons. And you can identify them, right? The C. elegans connectome and the neurons, we know all of them by name, so they basically used a microscope to pinpoint every one of the neurons. And then they took another worm species, P. pacificus, less well known, but interestingly, they could actually label the same neurons in the fairing system of the P. pacificus. Right? So we have two brothers from the same worm family, C. elegans and P. pacificus, which both have the same neuronal system. Now, C. elegans is a bacterial feeding worm, and P. pacificus is a predator. It can actually eat other worms. So the C. elegans is the vegetarian of the family, and the other one is the carnivore, right? And now they used, again, EM to reconstruct every one of the connections between the neurons of the fairing system in both species. And one particular behavioral difference between the two species, again, right, it's eating behavior, is that P. pacificus has a tooth-like structure. And this tooth is crucial for his predator type of behavior. Now, what Boombacher and colleagues found is that the wiring between the neurons, the same neurons in the P. pacificus, were much more centralized than one particular neuron PM4. Exactly that neuron that controlled the tooth-like dentricle. So crucial for the type of behavior that the animal showed. So to me, this really suggests that how the brain is wired shapes the functionality that it can display. And evidence for this type of theory also might come from studies more closer to home. Work done by Jim Reiling and Todd Preuss, in which they, for example, compared anatomy between humans and chimpanzees and macaques. And in particular, when they looked at brain wiring, they noticed that, particularly in the human brain, tracts like the uncinata fasciculus and the arcuata fasciculus are much more evolved in humans. Tracks that are really crucially important for higher order processing, in particular language. Again, suggests that the wiring structure might play an important role in shaping brain functionality. also in higher order animals. And even more, they also looked at network structure, and here too, both in structural and functional studies, we can observe that the hub structure across humans and chimpanzees might be, to some extent, overlapping, actually quite a bit, but there might also be subtle differences, with network hubs potentially more distributed across different cortical areas in humans, suggestive, as the authors suggest, of perhaps supportive, more complex and more diverse brain functionality. So from this, we could argue that perhaps there is a strong drive to form and to serve higher complexity in service of higher brain functionality, right, in evolution. Now, if this is true, again, we might also argue that perhaps that in the human brain, this drive to form more complex wiring and perhaps put more pressure on specific network attributes, again, being beneficial for higher complex brain function, this might also lead to perhaps some possibility that if there's damage to these systems, might lead to human unique type of brain dysfunction. And this is not a wild theory. This has been actually suggested already in the 90s, that perhaps a pressure on human brain evolution might have also brought some pressures to human unique type of brain disorders. And one of them in particular is, again, schizophrenia. Theories by Kroll and Burns, they've suggested that there might indeed be a pressure on human brain evolution, and in particular in service of social cognition and higher order language processing, that in turn might have brought a certain vulnerability, and it might have led to the development of schizophrenia. And evidence for this is, of course, the strong genetic background of the disorder, but also the human unique type of, it's unique to humans, right? It's not present, as they think, in chimpanzees. So this led to the argument that perhaps the disease actually started after the split of our common ancestor. So in a recent study, oops, let's save their privacy a little bit. I'm European. So in a recent study, we actually start digging into this deeper. So we took a population of chimpanzees and a population of humans, and we aimed to compare them as good as we could, right? Basically, we tried as hard as we could, just like in the worms, the Boombacher worm experiment, to keep the number of nodes and the regions exactly the same. So we did big effort to go into literature and look for cytoarchitectonic atlases with homologous regions across species. Many thanks to Lianne for doing this, as well as Rory, putting a lot of effort in this to be able to do this as good as we could. And then we used diffusion-weighted MRI to reconstruct the connectome in both species. And of course, we compared this. And this is obviously work to get it done with Jim Reiling, and a pioneer in comparative biology. And of course, first we found quite a big level of overlap between the two species, obviously, right? They are very alike, actually, in behavioral repertoire. But we also found differences. In particular, again, tracks around the ocular vesiculas and connections between language-brain regions, but also actually tracks to specific motor areas for higher order hand movement control, suggestive of support for higher order tool use as humans can display, as well as some tracks around social areas. And then we said, what if we would compare this to a map that we, for many years, have actually been examining, the map of disconnectivity in schizophrenia. So here we took a large population of patients and controls. Actually, we did 3 verification data sets here to be absolutely sure. And then basically for every one of the connections, we measured the level of disconnectivity. And when we compared the two maps, we noticed that in particular the tracts specific to humans, the ones that we could not observe in chimpanzees, were the ones that had the strongest level of disconnectivity. And even more, also the tracts that we could actually observe in both species, but the ones that were much more enhanced relatively across the connectome in humans were the ones that came second. So taken together, this might suggest that actually also variations in connectome structure might give rise to behavioral differentiation and perhaps leading to a quite unique type of brain functionality, but perhaps also brain dysfunctionality. So in the remaining time, I would therefore like to discuss this in a little bit broader perspective. So hopefully by now I've convinced you that these fundamental dimensions of the connectome are so crucially important for shaping not only the connectome, but also shaping with that brain function, right? And with that perhaps potentially the spectrum of brain function that we humans can display, and who knows, also what we cannot display. So if connectivity is so crucially important, and you all shout it out loud, so we have to assume, right? Then it is easy that things can go wrong. Right? Who knows? Perhaps it's almost inevitable that things can go wrong. And when we go back in time and we look at the last 5 to 10 years of connectome research, there is a growing number of studies that have reported connectome abnormalities in a wide range of brain disorders, right? Both neurological and psychiatric. I put 2 of them on each of this slide, but I apologize if your favorite disorder is not on here. But if you go on the Google, then and you type in your disease of choice and connectome, then very likely there's a paper about this. And if not, just ask your neighbor; he's likely working on it. So basically, this argues for all of these disorders to be involved, in all of these disorders to be the connectome involved, right? And I'm not into the like summing up all of these differences here for every one of those disorders, but actually, it brought me to, in the last year, to perhaps 2 big questions, at least for me, right, in modern day disease connectomics. And the first one, if it is really true that connectome is so important in brain function and in dysfunction, then is there perhaps a shared common substrate for disease in terms of connectome abnormalities? And that we should start looking not just at one disorder at a time, but that's actually what most of us, including me, have been doing, right? We just study schizophrenia or bipolar or autism or ADHD or ALS or Alzheimer. But perhaps we should start combining all of these studies and look for this shared common substrate. And second, and in a minute I will hopefully make some argumentation for this. Is it perhaps the case that the inherent organization, these dimensions that we've been talking about, might they actually be shaping also not just brain function, but also brain dysfunction? So the first one, right, the shared common substrate, was actually is actually the work of Simon de Lange, another brilliant student in the lab, who actually one day said, well, you've been looking at schizophrenia now, but how does this relate to the other types of disorders? So what he proposed to do is said, let's take as much data as we can, just reach out to our many collaborators, and we just collate thousands and thousands of MRI data sets. And then basically really not just in one disorder, but in this case across 12 different brain disorders. And we collected patients and controls of every one of them, and we did a disease connectome study, again, within every one of the studies, building a disease connectivity map for every one of those studies separately. And then we combined the disease maps. And we looked for which one of the edges of the network might be more affected, and in particular, we then said, what is the topological role of these edges? And what we noticed is that in particular the edges important for neural integration and neural communication were the ones that were more frequently reported across this wide range of brain disorders. And this led us to our second suggestion. Is it perhaps the case that these underlying principles of wiring are actually shaping the vulnerability to the brain? And it sure is apparently for the level of integration, right? So if we go back to our dimensional space, we have the 2 dimensions, right, with the trade-off in the middle, the blue line, and then at the 2 ends, we got the 2 different subspaces of network configuration types. On one, the ones that are biologically implausible, well, we don't have to look at them, they're not there anyway. But the green one is actually quite interesting, right? And then we said, if we have these 2 dimensions, how do actually the disease effects relate to these 2 dimensions? So first, let's look at the modular organization of the network. Well, there has actually been a number of series of studies now very recently proposed, in particular fueled by the pioneering work of Bill Seeley here, in which he noticed that if you look at the atrophy patterns of different neurodegenerative disorders, AD, behavioral FTD, and several others, then this atrophy pattern actually tends to overlap to known functional systems of the brain, and almost exclusively, right? So AD mostly relates to the default network, behavioral FTD mostly relates to the salience network. And from this, it has actually been now continuously proposed that perhaps the disease might start in a certain area of the brain and then spreads through means of a prion or perhaps functionally to other brain regions. But this spreading can only happen through means of the connections. Now, given the strong modular decomposition of the network, in the beginning, these disease effects are almost trapped within the neural module. So in first phases of the disorder, it's particularly the network that is primarily firstly affected, with the regions in that network that become mostly involved in the disorder. And this has now been suggested for AD, but also for Parkinson, and we did quite a fair bit of ALS in this. And actually, we used computational modeling in which we could almost one-to-one predict the level of spread of disease proteins to flow around the brain in ALS. So this suggests that the modular organization is heavily shaping the disease spectrum. Now let's look at the other axis, right? The one of the integration. Now this too might actually have a strong effect. In a review paper a couple of years ago, Olaf and I, we already noticed that these hubs are not only reported in one disorder, but actually there are papers popping up everywhere of hub involvement. So why is this, right? And this was nicely put forward and empirically validated by a paper by Crossley and colleagues, in which they did a large meta-analysis study in which they compared VBM analysis results from a wide range of brain disorders and noticed that in particular, indeed, the high level of degree regions of the brain are the regions that most often are reported in terms of disease abnormalities. So that argues for hubs to be quite beneficial for brain function, but also to form vulnerability hotspots. Now, hubs and the formation of global paths might also in another way bring vulnerability. In particular, the global structure of the brain might perhaps also facilitate fast cascading of effects. Right, the cascading failure theory of Alzheimer of Jones suggests that the disease might start in an area, and then this area becomes overheated, the areas around it have to take over, but then these areas become overheated, and then basically the disease cascades around the entire network. And it might be argued here that the global pathways of the brain might actually facilitate fast spreading of this effect. So let's go back one more time to our dimensional space. So we have now found evidence that perhaps these two dimensions might actually be shaping the effects that we can see in a wide range of brain disorders. So from this, Olaf and I, we started to argue that perhaps the human brain, right, it hovers around this optimal trade-off, but in the disease process, it is taken away from its optimal position into a suboptimal domain. It started to operate suboptimally. Now the point I want to make is that this transition is not random. It's not just a connection or a type of connection that starts to affect, and therefore there's a random process of going into the suboptimal domain. But this transition from the optimal to the suboptimal space is actually shaped and determined by the underlying dimensions of the human brain. So basically how the brain is wired can give us a lot of brain functionality, but unfortunately also brain dysfunctionality, and in a semi-ordered way. So in conclusion, I hope I convinced you that the brain is an extremely rich and complex network, and that there are principles of wiring, fundamental organizational principles that can give us brain function as well as shame brain dysfunction. I also hope that I have argued a little bit for a micro and macro bridge, some other passion of us in the lab, to actually underscore the importance of starting to link up these macroscale network features to the underlying neurobiology. Now, of course, I too have many people to thank, in particular, many collaborators over the last few years. I'm not going to give any specific names because that's never good, I think. But I should, of course, thank the people of the lab that actually are doing all of the work, and also our funded agencies. Thank you for joining.